I'm Will Parsons. I'm a pediatric oncologist at Texas Children's, and I run the precision oncology program here. And today I'm going to be talking about precision medicine, or more precisely, precision medicine for pediatric cancer patients. So what are we talking about when we talk about precision oncology? Essentially, we're talking about the tailor of cancer prevention and treatment based on the characteristics of each patient. So learning what we can about each individual patient and then tailoring our treatments accordingly. You can imagine all the ways that this might make sense, from the fact that we can make better diagnoses or more precise diagnoses and figure out what our patients' tumors are, to providing more useful prognostic information about the expected responses to treatment, to determine what the best treatments might be. And I'll talk a little bit more about how in this era of molecularly targeted therapies, we can actually identify mutations or changes in the patient's tumor that might give us clues about how they could be treated, both in terms of molecular targeted therapies as well as immunotherapies, in ways we're trying to activate the immune system to target a particular weakness in the tumor. And then last but not least, we can also identify potentially inherited aspects of this child's disease, either related to cancer or other diagnoses. This really builds upon a decade of discoveries in terms of the genetics of both pediatric and adult cancer. So this slide, for example, figures taken in a paper describing the discovery of genes of mutations in the isocitrate dehydrogenase, or IDH, 1 and 2 genes, in glioblastoma, the most deadly brain tumor that occurs in adults as well as in older children. And so this is a case where 10 years ago, IDH1 and IDH2 were not known to have anything to do with cancer. So there have been no mutations reported, there was nothing linking them to cancer biology. But by finding these mutations in a specific subset of adults and older children with glioblastoma, it identified both the molecular or diagnostic marker as well as a vulnerability. And over here on the right, you can see the crystal structure of this, where you can imagine targeting it with a small molecule or some other therapeutic to specifically target and treat patients whose tumors had this IDH mutation. And so now today, approximately 10 years after this being discovered, there are already FDA-approved therapies targeting IDH for patients with leukemia. So the whole premise of precision oncology is based upon the fact that cancer is in essence a disease of the genes, or a disease of the genome, meaning all of the genes. There's many ways in which we think about personalized medicine or precision medicine, and that's how we've been taking care of our patients as oncologists for years. I mean, what tumor do they have? How old are they? What other kind of features they might have that might affect their therapy? But here we're specifically talking about learning about the genetics and then understanding how that can impact patient treatment. And so on the left, you can see a schematic slide here showing a map which is meant to give you some understanding of, or some sense of, the complexity of human cancers. So this is a map of all the different mutations that have been placed on this plane, occurring in all different patients in a particular type of cancer. So no two cancers are the same. There's some genes which are mutated more often than others, these mountains. There's others that are much less common. But basically, it can be kind of a complex landscape, and no two children and no two children's tumors are exactly the same. So the premise for precision oncology is actually based upon that despite this complexity, there are a key number of activated biologic pathways. So some of them might be different in different patients' tumors, but this can be simplified down to particular genetic pathways that can be targeted with particular treatments. And that's the basis for what we're talking about here. We think of two different types of mutations when we're talking about precision oncology. The first and the most commonly occurring in tumors are somatic mutations, or tumor-specific mutations. And so these are changes that have occurred in a previously normal cell within the body. So a normal kidney cell, a normal brain cell, a normal skin cell that then acquired this change, and it's related to the development of the tumor. But then less commonly, there can also be germline changes, also known as constitutional changes or hereditary changes. And these are mutations that are present in many tissues of the body, potentially all of them, or a subset in some cases. And they predispose to the cancer, and they're also related to the development of those cancers. And importantly, those can be passed down from parents to their children. And so both of these types of mutations are relevant to how we take care of our patients. The last handful of years has been what I would consider the era of the pilot studies of precision oncology. And so this is an example of one of those from here at Texas Children's Hospital. So it's called the Basic 3 study. It stood for Baylor Advancing Sequencing in Childhood Cancer Care. And essentially it was a study meant to test this idea of precision medicine, the feasibility of it in essence. And so it had 2 goals, and the first was to use whole exome sequencing as one of these specific types of molecular tests, and to incorporate it into the care of our patients with solid tumors at Texas Children's Cancer Center. So to set up the laboratories to do that testing, to explain the risks and benefits of that type of test to our families, to interpret the results, to return them to the families and the oncologists, and to really understand how we could better apply precision oncology and how we could use it. And then secondarily, we were also hoping to understand what the utility of this might be. So to see what kind of tumor mutations we'd find that have an impact on how we pick treatments for patients. What type of germline mutations we might find that would explain why a particular child developed that cancer and might have implications for other family members. So over about several years, we enrolled 280. The goal was to enroll 280 children. We ended up enrolling close to 300 patients with all types of solid tumors, obtained blood and tumor samples, sent them to a clinical lab where they had this testing or exome sequencing done. Clinical reports, both a germline clinical report and a tumor clinical report, were generated. Those went back into the medical record, they were returned to the oncologists, and then our study team, including both oncologists and genetic counselors, would also explain those results to each family. And then we followed those patients. So in essence, a pilot study of precision oncology. And the first point that we learned was that it was feasible, that we could do all these things, and we could do it in a relatively time-sensitive fashion such that we give results back to patients in the course of their clinical care. This is an example of one of those patients. So it was a 14-year-old boy with a medulloblastoma, which is one of the most common brain tumors. You can see it's this white blob here back in the cerebellum or the posterior fossa. This is what it looks like under the microscope, just a sea of small round blue cells. And these are the results of the tumor sequencing, basically showing mutations. And on the top, we have the sequencing of the blood, and you can see there are no circles, meaning there are no mutations there. On the bottom, the results of the sequencing of the tumor. And you can see each of these cases, we've circled the mutations that were detected in that particular gene. Showing essentially for this patient with a medulloblastoma, in reality it's not just a medulloblastoma, it's a medulloblastoma with specific mutations in beta-catenin, making it a particular subtype of medulloblastoma, Wnt pathway medulloblastoma, a DDX3X mutation, which goes along with the beta-catenin, and then mutations in other genes such as P53, ARID1A, FOXO3 that have some implications for investigational therapies for these patients. So we could feasibly analyze the genomes in each of these patients in the clinical setting. And this is what that looks like at a much higher level. So going back to that same map, we found mutations in all sorts of key cancer genes that we know about, such as beta-catenin, P53, BRAF, and you can see right down the list. The key point, though, was that we found mutations in each of these genes in very specific populations of the patients. So for example, the most frequently mutated genes were BRAF, P53, and beta-catenin, and each of them was only found in less than 10% of patients. So if we had a drug targeting BRAF and you treated all of the patients, it would not probably make sense because the mutation is only present in a small number, a small but real number. And similarly, if you look at some of these other key genes, including some of these genes for which there are a number of investigational drugs available, they're really in very specific subsets of patients. So a strategy in which we treat all the patients the same way with the targeted therapy really does not make sense. The germline testing or the blood testing gives equally diverse results, which I won't talk about in any great depth, other than to say that we find mutations in all types of different categories that have potential implications for clinical care. So for example, finding mutations in genes related to inheritance of cancer or other diseases that the patient might have. The cancer mutations were found in about 8 to 10% of patients. We found mutations related to other diseases in a relatively small fraction. Mutations in other genes that we don't know that the patient currently has that disease, but it might make them more susceptible to that in the future. Those are in about 5% of patients. Genes that we call pharmacogenomic changes that affect how drugs are tolerated and dealt with, and then genes that are for recessive carrier diseases and other diseases where this might have some implication for either the patient or their children in the future. And so by sequencing on this large scale the patient's tumor in the blood, we can see the variety of all these different mutations that potentially have relevance for clinical care. So here's just a couple examples of how that can be useful. So this was a slide from a patient who had clear cell sarcoma of the kidney, so a relatively uncommon but very malignant kidney tumor. And we had one patient with that disease on our basic 3 study. When we sequenced that patient's tumor, we found this very odd type of mutation called an ITD, or an internal tandem duplication. This is a patient sequence here in each of these rows. Some normal part of the protein, or the gene, marked here in green, is duplicated or copied in an abnormal part, red. So instead of the gene ending right here, it has this extra sequence that gives it some abnormal function. We found it in a single patient, and then we went back into the Texas Children's Archives and sequenced other patients and essentially found very similar internal tandem duplications in almost every patient that we detected or analyzed. This has a lot of pretty practical implications. So the molecular pathologists here at Texas Children's have already developed a molecular test for this. So now if we have a patient with a kidney cancer and they want to know whether it's a CCSK or a clear cell sarcoma of the kidney, they can do a straightforward molecular test. It's also given us clues about the biology and inspired a lot of other research studies. We don't yet have treatments targeting this BCOR gene, though. This is another example of how this can be potentially useful. This is a patient with a type of sarcoma or a soft tissue tumor, and this is this giant tumor in the left lung or in the left side of the chest. This is what it looked like under the microscope. And when we sequenced this tumor, what we found actually was that it had what we call a fusion gene, or a gene that's an abnormal product of 2 different genes put together. In this case, a gene called BRAF, which is a very important cancer gene, and it's dysregulated or mutated or abnormal in a significant number of cancers, and that it had fused or copied with this other gene called KIAA 1549, giving a result which is not either one normally, but a combination of the 2, and resulting in a BRAF gene that's abnormally regulated and active and causing the cancer. This is very important for a couple reasons. One is that we would never expect to find this particular fusion in this type of sarcoma. This is a fusion that's very frequent in some specific types of brain tumors, and so if we were doing targeted testing of those tumors, we would have done that. But we would not have routinely thought to do it for a sarcoma. The other important point is that there are a number of drugs targeting both this gene and the pathway that activates, both FDA approved and in clinical trials. So it'd be a very unexpected result for this patient, one with potential therapeutic implications, and one where really making the case that doing broad genetic analysis of these patients is of some potential value. So that was the area of the pilot studies. And we've seen ways in which this type of testing can be useful for patients. We've seen that we can do it feasibly in the clinic. We've gained a lot of experience with how to explain these types of results to patients and their families, as well as clinicians, so that we can use them for research. So the question is, what are we doing now? And now we've entered the phase of testing how well this precision oncology approach works. So in clinical trials and in other clinical studies. And so this is an example of one of those. It's called the Texas Kids CanSeq study. It's a follow-up to our Basic 3 project where we were sequencing solid tumor patients here at Texas Children's. This time we're sequencing and enrolling patients from a larger number of institutions, so a handful of different sites, different size hospitals from around the state, with a real goal of doing this type of genomic testing, not just at a single big academic center like Texas Children's, but at a diversity of different institutions who have different specialties, as well as for diverse patients in terms of race and ethnicity, in terms of other factors that affect their treatment. And so the goal this time is not just to see whether precision medicine or precision oncology is feasible. We know it's feasible. We are proving it's feasible on a larger scale by doing it as part of a statewide study. But this time around, what we're really trying to understand better is what are the most useful tests? So not just is this broadly theoretically useful, but how can we best use this for our patients? And so what we're doing essentially is comparing a number of different types of molecular testing. So for example, for the tumor samples, we're comparing a targeted gene panel, so picking a selected number of cancer genes that we think are the most important ones for our patients, sequencing every patient's tumor with that, and then also sequencing every patient's tumor with much more extensive studies. So with exome sequencing, with RNA sequencing, with a copy number analysis, and putting all of that into a single clinical report, and really trying to assess what's the best way of doing this? How broad do we need to go? How quickly can this be done? How effectively can we find mutations both in the tumor and in the blood that have implications for these patients? There's also a number of questions regarding the feasibility of this type of testing. For example, whether we can return results effectively and safely to families by telemedicine or by other reporting means, and really a lot of questions that are designed around not just can we do this, but how can we do it best for our patients, both at Texas Children's and at other institutions. The other study, which we're very actively involved in, is another even more direct test of this precision oncology approach, and this is called the NCIOG Pediatric Match Trial. As you can remember, I mentioned that when we find the mutations in a particular gene, it's only in specific subsets of those patients, and that can be represented. So say this is the country, each of these stars is a patient and their tumor, and you can see there are all these different colors representing tumors that have different mutations or different changes in them. The goal is we want to match each patient's tumor to the drug targeting that particular gene that has the same color here in this diagram, for example. So as we have investigational therapies, not giving each of them to all of the patients, understanding that we only expect it to work in a very small number of patients, a specific number, but really trying to do what we can to sequence and analyze the patient's tumors to understand, well, who, in theory, what would the best match be? Which would be the patient that we would most like to study using a particular drug because we think it has the best chance of working. And so it's a trial for patients treated in institutions in the Children's Oncology Group, which is the collaborative group that does clinical trials all across the country, as well as elsewhere in North America and a few other countries around the world. And basically, the goal is to enroll at least 1,000 children with relapsed cancer, so all different types of cancers that have not responded well to therapy and who need some additional treatment. To obtain a tumor and blood sample from those patients, to do this testing, to see what mutations or changes we can find, and then based upon kind of predetermined levels of evidence, matching up particular mutations to particular drugs, figuring out if we have a match from a whole menu of different investigational agents of targeted therapies that we could potentially give that patient that we think might have a chance at offering some benefit. So we do that, we match patients up to targeted therapy, and then they receive that therapy in standard phase 2 clinical trials. And so it's really set up as a screening protocol and then matched treatment protocols for a subset of patients. And so we're so far have 10 different drugs in this menu, and the idea is to continue to add different drugs as new exciting drugs become available to maximize the probability of finding a match for any individual child. And then testing both the feasibility of this approach, what we can find when we do this sequencing, how many patients can we actually match up to one of these drugs, and then most importantly, do those drugs actually offer some benefit for our patients? So this is the next extension of the precision oncology approach is not just can we analyze patients' tumors in their blood and find mutations, but can we actually do something about it that offers benefit to our patients? So in conclusion, I hope I've conveyed a couple of key points. The first is that there's a lot of excitement, enthusiasm, and potential for precision oncology approaches for pediatric cancer patients. The important mutations that we find are often in specific subsets of patients, and they're often in unexpected patients. So we cannot always guess which patient, based on their tumor type or how old they are, might have a particular mutation that might respond best to a particular therapy. We have a number of studies ongoing to determine how to best implement this type of precision medicine approach, from explaining the risks and benefits and potential results to families, to counseling clinicians on how best to use those results to make decisions for their patients. And finally, we have a number of prospective clinical studies that are currently underway testing how this can offer benefit to patients at Texas Children's and around the country. So we'll thank our patients, and I'm happy to take any questions after we're done. Thank you.